Welcome back, everybody. In this lecture, we'll be talking about the examples of protein functions and applications in continuity. So, now we'll be talking in detail about the membrane proteins. As we all know, the antibodies have similar structure and function. Enzymes, although they are structurally varied, have a common function, that is the catalytic function. However, the membrane proteins are slightly different. All the membrane proteins are definitely located at the membrane, but they may be structurally and functionally diverse. As we know, the biological membranes have the same basic phospholipid bilayer structure. However, each membrane has its own set of membrane proteins that give these membranes their distinctive activities. The complement of proteins attached to a membrane varies depending on the cell type and subcellular location. So, this basically depends on the cellular function as well. And some proteins are bound only to the membrane surface, whereas others have one region buried within the membrane or with the domains on both the sides of the membrane. So, with this, let us try to know the different types of membrane proteins. The first type of membrane protein is the extrinsic or peripheral membrane protein. There are two varieties of extrinsic membrane proteins. The first variety is known as the extracellular membrane surface protein, that is present on the outer side of the cell membrane, meaning facing the extracellular matrix or the neighboring cells. And this kind of a membrane protein is involved in cell signaling or interactions. The next variety is the intracellular membrane surface protein, which is located or which faces the cytosolic phase of the cell. And it has got a wide range of functions that can range from anchoring cytoskeletal proteins to the membrane to triggering the intracellular signaling pathways. The other type of membrane proteins is the intrinsic or integral proteins that form channels and pores to facilitate movement of molecules across the membrane. So here in this diagram, as you can observe, this is a phospholipid bilayer or a section of a cell membrane where you can see that there are integral proteins, there are peripheral proteins, and all of them are either facing the exterior of the cell or the cytosol, etc. Now, trying to understand the protein membrane interaction with reference to intrinsic proteins. The integral membrane proteins have one or more segments that are embedded in the phospholipid bilayer, and they contain residues with a hydrophobic side chains, and they interact with fatty acyl groups of the membrane phospholipids in order to anchor and span the entire phospholipid bilayer. Additionally, there are another variety of membrane proteins which are known as the transmembrane proteins, which have one or more membrane spanning domains as well as domains from 4 to several hundred residues long that extend into the aqueous medium of each side of the bilayer, meaning that they are, they start at the extracellular phase of the membrane and they move all the way up to the intracellular phase of the membrane or the cytosolic phase of the membrane. Additionally, there are integral proteins that are anchored onto one of the membrane leaflets by covalently bound fatty acids. And this bound fatty acid is embedded in the membrane. But the polypeptide chain does not enter the phospholipid bilayer at all. However, the protein membrane interaction in the case of extrinsic proteins is slightly different from that of the intrinsic proteins. The extrinsic proteins do not interact with the hydrophobic core of the phospholipid bilayer, and they are usually bound to the membrane directly by interactions with the integral membrane proteins or by interactions with the lipid polar head groups. These peripheral proteins are localized to the cytosolic phase of the plasma membrane. For example, there is the cytoskeletal proteins such as spectrin and actin in the case of erythrocytes, and also there is a enzyme protein kinase C, which shuttles between the cytosol and the cytosolic phase of the plasma membrane in order to fulfill its role in signal transduction. There are a few other peripheral proteins, including certain proteins of the extracellular matrix or the ECM, which are localized to the outer surface of the plasma membrane, that is also known as the exoplasmic surface of the cell. Now, moving on to the other variety of membrane proteins, these are called the receptor proteins. And these are the protein molecules that may be present inside a target cell or on its surface with the only purpose of receiving chemical signals. These chemical signals are generally released by the signaling cells and they may be small, volatile, or soluble in nature. And they are generally called ligands, where the ligands are the molecules that bind to another specific molecule, in this case, a receptor protein, with the purpose of delivering a signal in the process. The ligand receptor interaction is specific, meaning that a specific ligand will have a specific receptor that typically binds only to that ligand. So now trying to understand the different types of receptor proteins. There are two types of receptor proteins. The first type is the internal receptor protein and the second is cell surface receptor protein. Getting into the details of internal receptor protein or the intracellular or cytoplasmic receptors which are found in the cytoplasm of a cell. They typically respond to the hydrophobic signaling molecules or the signaling molecules that can bypass the phospholipid bilayer or the cell membrane and enter into the cell. Once inside the cell, many of these signaling molecules bind to proteins that act as regulators of mRNA synthesis. And when the ligand binds to the internal receptor, there is a change in shape or conformation of this complex, and this triggers an exposure of the DNA binding site on the receptor protein. And now the receptor ligand complex moves into the nucleus and binds to the specific region of the DNA to promote the production of mRNA from those genes, thereby indicating that the internal receptors can directly influence gene expression without having to pass any signal onto other receptors or membranes or messengers. Cell surface receptors, the second type of receptor proteins, are usually the transmembrane receptors that are found attached to the cell membrane, and they bind to the external ligand molecules which are not necessarily hydrophobic in nature. This kind of a receptor spans the plasma membrane and performs a signal transduction where an extracellular signal is converted into an intracellular signal, thereby implying that the ligands do not have to enter the cell. The cell surface receptors, which are also called cell surface proteins or markers, are specific or unique to individual cell types. And there are three main components in a cell surface receptor. The first is an external ligand binding domain, which is followed by a hydrophobic membrane spanning region, and then comes the intracellular domain inside the cell. The size and extent of these domains vary depending on the function of the receptor and also the type of the receptor. There are three types of cell surface receptors. The first variety is called an ion channel linked receptor, which binds a ligand to open a channel through the membrane to allow the passing of specific ions from the outside of the membrane to the inside or from the inside of the membrane to the outside. And such ion channel linked receptors have an extensive membrane spanning region, and upon binding of the ligand, these membrane spanning regions undergo a conformational change in order to create a route through which ions like the essential ions like sodium, potassium, calcium, or magnesium can move across the membrane. The second variety is the G protein coupled receptor, which binds to a ligand to activate a membrane protein called the G protein. And this activated G protein interacts with either an ion channel receptor or an enzyme in the membrane to carry out further signaling mechanisms. However, prior to the ligand binding, an inactive G protein can bind to a site on the specific receptor as well. And once the G protein binds to the receptor, it undergoes a change in conformation, becomes active, and splits into two different subunits, which may be able to activate other proteins involved in the signal transduction. Finally, we have the enzyme linked receptors, which are cell surface receptors that are generally possessing an intracellular domain that is associated with an enzyme. In some cases, this intracellular domain of the receptor may itself be an enzyme, whereas other enzyme linked receptors may have a small intracellular domain that interacts with an enzyme directly. So, whenever a ligand binds to the extracellular domain of the receptor, a signal is transferred across the membrane to activate that enzyme associated with this receptor, and this activation of the enzyme sets off a chain of events within the cell that will eventually lead to a response. A classic example for such enzyme linked signaling is insulin, where the insulin receptor is an enzyme linked receptor. And if you have any doubts, you can always contact me. Thank you.